Joining me is Dr. Eva Chi, Assistant Professor of Chemical Engineering at the University of New Mexico, doing research in the area of protein dynamics. And more specifically, I guess one of those topics, Dr. Chi, is Alzheimer's. What is preventing us from finding a cure?
We don't yet have a good understanding of what causes the changes that we see in Alzheimer's disease. The healthy brain is made up of billions of neurons in a really interconnected network. In an Alzheimer's disease brain, a lot of the neurons have died, and so the connections have been lost. And in its place, what we see are the dead neurons containing neurofibrillary tangles inside the neurons, and in between the neurons, we see these amyloid plaques. And what happens is that this causes brain degeneration. It disrupts the neuronal network. So a lot of the brain functions are compromised.
All right, so let's look at a healthy brain, for instance, and what are we seeing here?
So the brain is made of billions of neurons, and all the neurons are interconnected, and they make trillions of connections.
You talked about plaques and tangles, and those are some of the things that you see in the diseased brain.
Exactly.
And that's a much different, that's a big contrast compared to the healthy brain. Talk a little bit about that.
Right. So in place of healthy neurons, in Alzheimer's disease, there's a lot of neurodegeneration, meaning that a lot of these neurons have died all over the brain, and the brain as a whole literally shrinks in size if you compare Alzheimer's brain to that of a healthy brain. In place of that, there are two prominent hallmarks that we see, which are these neurofibrillary tangles inside dying or dead neurons, and also plaques, these massive proteins, amyloid plaques, that are in between the neurons.
What makes the good plaques go bad? Do we know that?
So what we know is that these plaques are composed of protein aggregates. That is, these proteins have come together to form in a particular morphology, particular form, in that they come together to form these fibrils. And that we know that both the plaques and tangles are composed of these tangles. What we don't know is what caused these proteins to undergo this transformation of forming these fibrils. So there are two questions that we still don't have the answers for. One is why do these proteins, in the population who does get Alzheimer's disease, undergo this process to form these fibrils, and also during the process, what causes the neurons to die? Why do these proteins start to lose their their good structure to form this structure that are found in diseased brains, and what's the process of forming that? So what triggers it, and how does the process actually come about?
Do we know how we prevent them from losing their structure or their health?
We know what keeps the protein happy.
What's that?
In that
what keeps it happy and looking like that first slide we saw?
Right. So proteins, their functions depend on their structure. But these structures are very delicate. So you can think of the protein structure as a house of cards. So if any disturbance happens to any part of the protein, then the whole structure can fall apart. So it is that delicate. And actually our body spends a lot of energy to keep, for example, our body temperature at 37 degrees, keep the pH and water content inside and outside the cells exactly the same. We spend, like I said, a lot of energy maintaining that. So we know that under those conditions, proteins are happy. We know that the structure of the protein can become destabilized. And one of the things that happens is once they destabilize, they can become very sticky towards each other, that they start to form these aggregates. If you think about, again, the analogy of house of cards, once one piece is removed or even bumped a little bit, then the whole structure falls apart. So what we're trying to do is find that trigger.
And do you feel like you're making some headway in your research?
Yes, we are.
Really?
So I had mentioned a few things, such as pH temperature can affect the structure of the protein.
Right.
And those things obviously don't change very much in the brain because, like I said, we are very good at maintaining the homeostasis.
You mean we as humans, we're actually
humans.
pretty good at keeping our body temperature?
Exactly.
Okay.
Yeah. So what we're trying to do is, what about other factors?
Sure.
So basically in my lab, we're trying to study these proteins. We're not yet working with brain tissues or with patients, but what we're doing is we're taking that protein and we're kind of simulate the environment that it would see and see what kind of changes can trigger the initial change in the protein.
And do you feel like there's anything specific to that you've learned now from simulating that environment?
Yes. So our hypothesis has been that these proteins, in particular the ones that are found in diseases, and again, what we're studying applies to not just Alzheimer's disease, but this class of amyloid diseases in general,
which includes Parkinson's and a host of other diseases.
And Huntington's disease, for example.
Okay.
So what we believe is that there are some commonalities with these proteins, in that they are, I guess, promiscuous in their interactions, that they have a higher tendency or propensity to interact with other structures in the cell. So what we're trying to explore is really kind of their abnormal interaction with the cell membrane that maybe can trigger the protein to what we call misfold, meaning that they go into a structure that's not a native structure.
Do they break the membrane of that cell they're in? And
that's a really good question.
Okay.
So the second line of investigation that we're trying to look at is to answer the question of toxicity. Why do these become toxic, or how do these proteins exert toxicity during aggregation? So what we're trying to do is looking at interaction between these proteins with the lipid membrane, and how that interaction affects the structure of the protein, and also how those interactions affect the integrity of the membrane.
I mean, this is fascinating research. How do we actually see this kind of research?
So what we do is we look at proteins on different scales. And what we do is we do a lot of microscopy. That means microscope, fluorescence microscope, where we have molecules. So we're looking at proteins on a molecular level. One of the instruments that we use is a Lamier trough with a fluorescence microscope. So there we can form a lipid membrane and then see how it interacts with the protein. We can measure the interaction precisely. And then we can also look through the fluorescence microscope to look at how these interactions are actually affecting the structure of the membrane. So that's kind of on the micron scale. 1 micron is 1 × 10⁻⁶ meters. So it's very small.
Nice.
And then from there we can look at angstrom scale, which is 1 × 10⁻⁻10. And we use transmission electron microscopy, for example, to look at the structure of the fibrils that are formed under different conditions. And then we also use X-ray and neutron scattering to look even in more detail on the molecular scale, kind of the structure of the protein and structure of the membrane. And so for those experiments, we go to Germany to a synchrotron source to do our X-ray scattering experiments. And we also travel to Los Alamos and collaborate with our scientists there to basically use X-ray or neutron to hit the membrane and the protein. And then so from there, we can resolve really angstrom scale structural detail of the proteins and the membrane that we're trying to study.
And what about the students? Do they get to partake in this type of research at that level?
Yes. Well, the students actually do all the work.
Oh, wow.
So my graduate students are the ones who are in the lab, using the trough, using the microscope, and they travel to Los Alamos, for example, to do the experiments. They do the data analysis, and then of course we talk about where the research is going, and we sit down and look at the data to see what it means. And so there's a lot of interaction between my students and I, but the students are actually the ones who does all the work.
What have we learned? What have we learned from this kind of research this far?
So what we've learned is that in the presence of certain lipid membranes made of certain lipids, they form the fibers much more quickly and at much lower concentrations. And the second thing that we found with their interaction with the membrane is that once these proteins start to aggregate, they interact more strongly with the membrane, and they can actually destroy the integrity of the membrane, essentially making the cell membrane that encloses the cell leaky. Mm-hmm. And once that happens, the cell undergoes, the cell can die. So your question about what can we do about it, what we envision is if the interaction between these proteins and the cell membrane can trigger or seed structural changes in the protein such that they undergo aggregation to form these fibrils, then if we block that interaction between the protein and the membrane, we can potentially prevent these fibers from forming.
And then based on that, do you see that this is where science is going with this research?
Well, so in terms of Alzheimer's research, as you would imagine, there's a lot of research going on in the field. When a disease happens, it doesn't really care, what realm of science.
Right. It doesn't discriminate.
It doesn't discriminate. So it really takes the whole scientific community to learn more about these problems and to hopefully come up with an answer.
You would have to feel like what you're doing is actually leading to that bigger picture of a cure someday.
Yes, I do. I think, I see where my ultimate goal is. I would like to contribute towards a therapy that can either stop or reverse the changes that happen in the brain that cause Alzheimer's.
And can that happen in your lifetime, do you think?
Oh, I think so. Yeah. What I'm driven by really is kind of the fundamentals. I want to understand how proteins behave. Some of my collaborators here at UNM are working on developing other engineering applications where proteins are at the heart of all these technologies. For example, biofuel cells, where we're engineering proteins in our body, enzymes in the Krebs cycle, to put them in a fuel cell that can use sugar essentially for energy.
Wow.
And convert that into water and CO2. Another area are biosensors. So think about tests on a paper strip. That you can test for water quality, for example, test for toxins, test for diseases.
Mm-hmm. How much do we really know about Alzheimer's, and how close are we to a cure, in your estimation? That's the big question, right?
That's a hard question to answer because the disease is really multifaceted. And like I said, researchers in different fields are looking at this problem.
Do we know a lot more than we did even 10 years ago?
Oh, yes, we do.
We do?
Yeah,
okay.
we do.
How about 5 years ago?
Yes. Yeah, there's just a lot of effort and resources being put into this field because it is such a big problem. Sure. And it's going to become a bigger problem as the population ages. Mm-hmm. Unfortunately, there hasn't been a silver bullet, and part of the problem is, again, we don't really know what's actually causing the disease.
People tend to want to feel like they have some control. Mm-hmm. Are there ways that we can keep our own proteins in our brain from misbehaving, or we can keep our brains healthy, or at least a little bit of prevention? Are there ways to do that?
Yes, I guess the answer is yes. I guess the two risk factors that we know for Alzheimer's disease, one is aging.
We can't stop that.
We can't really do too much about that. And second is there are some genetic links to early onset Alzheimer's. But for the vast majority of patients with Alzheimer's, 95 to 98%, there's really no hereditary cause or genetic cause. So for the late onset cases, if you look at on the genetic level, there's really no difference between the population who gets Alzheimer's disease versus the people who don't. So in those cases, there are some general things that we can do to, basically keep our body healthy, keep our brain healthy.
What are those things?
Well, this may sound cliche, but eating a healthy diet, keep an active lifestyle. So exercise not only does the body good, but there's been multitude of research to show that exercise actually keeps the brain, is very, very good for the brain.
Challenge yourself, like do crossword puzzles and different things, or read, or stay active, look at...
Mm-hmm. Yes, stay active, and I'm sure that's what your doctor tells you as well.
Sure.
Is when people age, if you keep the brain active, Then in terms of it improves memory, it improves cognitive abilities, which are all the areas of the brain that are affected by Alzheimer's disease. There is a correlation between, kind of the Western high cholesterol, high fat diet versus the incidence of Alzheimer's. And there's also tend to be a higher incidence of Alzheimer's in women compared to men. One suggestion has been that women tend to accumulate more oxidative stress, particularly in their brain as we age compared to men.
And is that how we keep a healthy brain, is not to accumulate oxidative stress?
Yes.
How do we not do that? It's all about keeping the brain healthy.
Well, the things that we talked about, healthy diet and active lifestyle, I think those all contribute to
a healthy brain.
Mm-hmm. We don't really have targeted therapies to slow down the progression of Alzheimer's or even reverse the process.
Right. We still have a long way to go in that area, but we do know there are certain things we can do to keep our brains healthy.
Right. Yeah.
Thank you.